So good morning. Let's see if I can get this to work. Great. So just as a quick overview on the company for you who are not familiar with Altogen, we're a company located in Research Triangle Park, founded in 2001, venture-backed company. The original technology that we licensed in came from Duke and Johns Hopkins. It is a unique way of isolating adult stem and progenitor cells from various tissue sources. And it's based on using an intercellular marker called aldehyde dehydrogenase, or ALDH, to use that activity to actually sort out and isolate stem cells. There's a lot of literature. I'll talk a little bit about the literature, but this is now known, I think, pretty well to be a marker of stemness. And we've basically incorporated this isolation technology into a full suite of GOPs or SOPs that allow us to manufacture cellular products that we call ALDH bright cells. Those are cells that express this enzyme. We are focused on a range of cardiovascular indications. We have a product, ALD401, for ischemic stroke, and I'll spend some time talking about that program. We have actually completed a phase 1/2 study in critical limb ischemia with the ALD301 product, and we've completed a phase 1/2 study in ischemic heart failure with our ALD201 product. I'd like to say we're starting a hepatitis C program soon because of the markets, but that's still on the block. So our approach is an autologous approach in the cardiovascular area. It is unique in a couple elements. So we're using a patient's bone marrow as the source. We ship that bone marrow to our site. We take about 150 mLs of bone marrow, which can be done in an outpatient setting. We then isolate out these ALDH bright cells, do full product characterization. That's one of the nice things about this technology is it's not only a marker for allowing us to isolate the cells, but it's actually a full characterization tool. We can do purity, identity based on this expression levels. And then we send those cells back to the clinical site where they're delivered locally for these indications. The time frame from when we receive the cells to the time they're back in the patients, anywhere from 24 to 36 hours. So it's a very rapid autologous approach. We don't culture, we don't manipulate the cells. And so from an ability to scale and also do have very reasonable cost of goods, we think this is a very efficient autologous model. So in 2001, we had a couple really key achievements. The first was that we got FDA clearance and began a study of ALD401 in stroke. We took the tack that we wanted to go very quickly into a study that would have some meaning. So we got clearance for a 100-patient, phase 2, randomized, double-blind study. And we've got a lot of the key opinion leaders in the United States behind this. Dr. Sean Savitz, University of Texas, one of the key thought leaders in cell therapy for stroke, is our lead PI. And there's a couple unique things about this study, I think, beyond the fact that it is probably the largest stroke study with cell therapy underway. We have started enrolling in this study. So a couple unique things. One is that we're going to give these cells 2 weeks after the stroke. There's some biological rationale there. We want to give the cells after the acute inflammatory cascade, but still while the brain still has some plasticity, and so that's where the 2-week time frame came from. But it also has unique aspects from a clinical development and logistics perspective. We don't have to worry about getting patients into the ER within a certain number of hours, so we've got a 2-week window there. And the other benefit, and one of the reasons a lot of the stroke studies have failed in the past, is they've looked at the acute window, and there's still a lot of variability in many of the measures in this very early time frame, so you get a lot of noise. So you need to do big studies to be able to filter out that noise. By going out 2 weeks, most of these patients have stabilized in many of the NIH stroke scale and modified Rankin score scale, so we can get a much better population that we think where we can show an effect. So I mentioned we're enrolling patients right now. The primary endpoint we're looking at here is obviously not only safety but modified Rankin score. It's the endpoint the FDA will accept for approval of a product in stroke. And we believe that the 100-patient study is powered enough that we can get enough signal that we can then adequately power a phase 3, and that was the intent of the study design. The other couple other achievements in 2011, we actually now have about over 250 publications on our technology, so this is a well-vetted technology. And we were the first company to actually publish clinical data in critical limb ischemia with cellular therapy, and that came out in October. And we did have a publication of our heart failure study accepted in the American Heart Journal, which would be published in the first quarter. So one of the first companies to really get out this clinical data with cell therapy into the literature. Now, rather than go through some of the data in CLI and heart failure, which is out there, what I thought I would do is focus a little bit on the question that we get a lot, which is how do these cells work? And I think a lot of people in the field now are really getting a lot more questions about mechanisms of action, having to address that not only with investors but obviously with regulators. And our view is that the ALDH bright cells from bone marrow have two primary mechanisms of action. One, they're very efficient at homing to sites of ischemia. And once they're there, they have a very powerful paracrine signaling ability that we think is important for tissue repair and regeneration. There's a lot of models out there for how cells home to ischemia. You can see the diagram here on the slide, but it's known that MSCs, EPCs, et cetera, home to areas of ischemia using integrins and selectins, and I'll show you some data as to what our cells express. And then obviously there's a lot of data in terms of what paracrine molecules might be important in angiogenesis and neurogenesis, and I'll show you some of that data on our cells. So related to homing, we've actually done a number of models where we've labeled these cells, and you can see here both in the hind limb model, the AMI model, and also the stroke model where we've labeled these cells, and you can see they're very efficient at homing to the site of ischemia. And then the question is why? And so we looked at these integrin expression and selectin expression on our cells. You can see that there are a number of markers that are highly expressed in our cell populations that really lead to this ability for our cells to home. And these are some of the markers that we will be studying in our phase 2, phase 3 studies to see if there's an indication that whether or not the levels of these expressions actually might correlate to clinical benefit. Then moving on to the paracrine side of things, we've actually now done an analysis, and this was data that was published in December, that the ALDH bright cells from bone marrow express over 60 pro-angiogenic and neurogenic genes that are very important. In terms of the angiogenic molecules, we know that these cells are able to promote the endogenous endothelial cell migration that's important for new vessel formation and vessel sprouting. We also know they're important for increasing capillary density. And the cells also express neurogenic molecules that are important for neuronal differentiation and synaptic plasticity. So to the earlier point that we heard, they're multimodal. They can do multiple things in the context of this disease. And I think with stroke, again, it's important to realize it's complicated, and I think it's going to require a cellular therapy to address those issues. Just a little bit of data which we think is interesting from a paracrine signaling perspective. We built an in vitro model looking at tubule formation using HUVEC cells that are cultured on Matrigel, and you can see on the left that's the baseline assay. We then put that assay into a hypoxic condition. You can see that those tubules then are diminished over time. And then we put our ALDH bright cells in transwell, not connecting to the HUVECs, but atop them. And it was clear that those ALDH bright cells were able to express soluble factors that then allowed the HUVECs to maintain their capillary or their vessel formation and actually enhance their formation. We also saw that these cells differentially express new genes based on that signaling cascade from the HUVEC. So this was a good proof to us that, again, these cells have very powerful paracrine signaling capabilities. The other thing that this actually showed us when we looked at gene expression was, why do you sort these cells? I mean, people have been using bone marrow themselves. Why not just give bone marrow? So what we did, if you look on the left, this is the hind limb model. Vehicle at day 21, you can see not much restoration of blood flow. If you put whole bone marrow into the model, you get a little bit of a benefit, but not a great deal. If you go to the bottom, if you put the ALDH bright cells, you get almost full restoration of blood flow. Then if you take what we call the ALDH dim cells, that's everything else in the bone marrow other than the bright cells, and put them in the model, you can see you get no activity. So we say, well, why does that happen? And why is bone marrow not as good as bright cells? So if you look at the gene expression of the dim cells, they don't express the things that the bright cells do that we just showed you, the angiogenic factors, the neurogenic factors. What they express are a number of these inflammatory molecules. And so the belief is that those cells, if given with the bright cells, are going to be in the way of homing, in the way of this paracrine signaling capability. And so it's important to take the bright cells away from the dim cells, and that's where you get the enhanced activity. And it's through these mechanisms of looking at MOA that we've been able to explain why to sort these cells out and why that is beneficial. The The other interesting part is that it's nice to see this data in preclinical models, but can you actually make the connection to the clinical data? So we know in the in vitro model I showed you, we can make these new vessel formations. We know that are active in models of ischemia. What happens when you get into the clinic? And it's been nice because in both the critical limb ischemia and the heart failure studies we did, we saw two things. One is, by different measures, the patients that got our cells actually improved perfusion in the areas that we put their cells. So in the limbs of ischemia, CLI patients, we saw improved blood flow, and in the hearts of the heart failure patient, we saw improved perfusion by a SPECT. So that was a connection, again, to our mechanism of action. And then the other thing we saw was that in all these patients where we had improvement in perfusion, we actually saw a clinical benefit. So we saw with critical limb ischemia an ability to reduce pain and improve patient outcome. And then in the heart failure setting, we saw an improvement of max VO2 or exercise capacity. So now we've made, I think, the connection from preclinical data through to the clinic that really validates our proposed MOA. So for 2012, our milestones are numerous. Hopefully we can hit them all. But with ALD-401 Phase 2 study, we will complete the safety phase of the study, which is a roll-in of 10 patients. We will do that in the next month or so. And then get near completion of the 100-patient study by the end of the year. So this is a study where we will have readout on the modified Rankin scale changes at 3 months in the first quarter of next year. So within 12 months from now, we will have data in a major Phase 2 study in stroke. We also plan to start a Phase 1/2 study in PAD, will not be in CLI, and also another Phase 1 study in a new CNS repair indication. Both of those studies, I should say, will be funded by third parties, so I think it's nice validation for our technology. I mentioned we'll have a publication of the heart failure data in the American Heart Journal. And then I think importantly, we're going to continue to publish some of this MOA data that we've been developing. I think it's where this field needs to continue to evolve so that everyone has the best idea how these cells work. And then clearly one of the hurdles we'll face is the potency assay for ALD-401. But I think by understanding that mechanism of action, we've got a number of things we're looking at in the trial that should help us put that together. So thank you for your time.